So this is going to be a bi-monthly, so twice a month, lecture series going until about May, and it's driven by the C-STAR project. So the C-STAR is the Center for the Study of Aphasia Recovery. It's funded by the NIH through a P50 grant and started in April. It's a collaboration between researchers here at the University of South Carolina, led by Julius Fridriksson, who we'll hear about later on, well, who we'll hear from a little bit later on. Researchers at Johns Hopkins University, R.T. Hillis, University of California at Irvine, Greg Hickok, and researchers at the Medical University of South Carolina, Leo Bonilla, who we'll be hearing from next week. All the talks and the research projects in the center are about aphasia recovery. So what we're trying to do is to get a better grasp of the neurocorrelates of recovery after aphasia, but also behavioral intervention methods. What's the best way to get at this? And Julius will be talking about that today as well. He will give an overview of the center and the different research projects that are going on there. The meetings are can be attended, as some of you are now, via GoToMeeting. We will always use the same link, so every time we have a new lecture, you can just log in with the same link and access the presentations online, live. We will also make a recording, which after post-editing we will put up via the C-STAR website, which is online, I think, as of today. And the website is there on your screen. It ends in C-STAR.
So I'll send you instructions for how to do that.
So coming up not next week, in 2 weeks, September 22nd, we'll be hearing from Dr. Leo Bonilla, who is at MUSC, and he will be talking about the human-brain connectome applied to stroke rehabilitation research. That's part of the contribution that he makes to this much bigger project, and I'm sure he'll be talking about some wider aspects of his research as well. Today, however, without further ado, I want to present to you Dr. Julius Fridriksson, one of our own, and he'll be talking about the research projects involved in the C-STAR Center. If people can't hear me from abroad, GoToMeeting, please give me a little chat. I'll be monitoring that. I'm also, for those of you who are watching this online from a distance, I'm going to shut off webcams from outside other than the webcam pointing at Julius, and I'm also going to mute all other microphones except for Julius's microphone. We will have a question session after the presentation, and then I'll just open it up. Otherwise, it gets a little bit confusing with all the people, talking from different parts of the world. So, Julius Fridriksson.
All right, thank you very much, Dirk. So, yeah, it's great to have all of you here. I appreciate you showing up. Also, for everybody who's online, I'm a little bit surprised how many people we've got in the attendance so far. I think it's 29 via online. Certainly our goal is to increase that number. I think we have up to 99 access licenses for GoToMeeting. I also want to say that ultimately what we're planning to do is we're videotaping the lectures. So in the future, these lectures are going to be available on the C-STAR website for anybody who wants to view them. Dirk has done a really good job of coming up with a very good lineup of speakers. So what we thought might be appropriate is that I give you a little bit of an overview of our new center, since the lecture series is associated with C-STAR. So just a little bit here on the background. So what is it? It's a research center focused on improving recovery from aphasia. It's funded by a P50 grant from the NIDCD, or the National Institute on Deafness and Other Communication Disorders. It's a multi-site center, like Dirk said, being done here at USC, Johns Hopkins, University of California, Irvine, and MUSC, or Medical University of South Carolina in Charleston. It's a 5-year grant, just started earlier this year and will end in 2021. Since we think there are going to be people attending who don't know aphasia that well, we thought we would start with a very brief background on aphasia. Aphasia is a language problem caused by damage to the language regions of the brain, usually the left hemisphere. It's typically caused by stroke. So what we see there is a couple of brains, those darker areas that you can see here with a cursor. These are areas that were affected by stroke, are now missing, and as a result, the person has some kind of a behavioral impairment. There are two types of stroke, either hemorrhagic or ischemic, that is brain bleed or an artery blockage. People can also have a result of things like brain tumors, traumatic brain injury, but what we are focusing on in C-STAR is strictly stroke-induced aphasia. There are different types of aphasia, and they very much depend on the patterns of brain damage. This is a figure from a paper from last year by Grigory Yorganov, where we showed that you can predict the type of aphasia that a person has simply based on the neuroimaging, the brain scans. And you can see that the different types of aphasia there along the left margin. For example, conduction aphasia, the best predictor of that type of aphasia is simply damage here in the temporal parietal junction, and this has been shown classically. But what we're trying to show there is that the different aphasia types are associated with very specific patterns of brain damage. Aphasia affects the patient, certainly, the family, and also friends. I included a figure there on the right. This is from an article in the New York Times from earlier this week that suggested that loneliness is one of the better predictors of early death, or early death in older adults. I think that with people with aphasia, that window is moved up much further in time, because aphasia certainly doesn't just result in problems for the patient itself, but also has problems for the family, friends. People with aphasia very often complain about that they don't have the same social connections that they did before. It certainly affects economic status, things like education and rehabilitation. And we would contend that the ability to communicate is a basic human right. So as we try to restore somebody's ability to talk, we would think that we're trying to restore somebody's part of their, or something that's a part of their dignity. Aphasia statistics, there are about 700,000 new strokes in the USA every year. It's thought to be the #1 cause of serious adult disability. Approximately 20 to 30% of cortical strokes result in aphasia, typically involving the left hemisphere. And it's estimated that 1 million people in the USA have aphasia today. For all patients with acute aphasia, so let's say that somebody had a stroke yesterday or the day before, about 75% of them experience some degree of recovery, even without any kind of intervention. But a quarter of them actually do not get better, and in many cases, those people actually get worse. Once aphasia is chronic, so let's say 6 to 12 months post-stroke, the greatest chance of recovery is via aphasia therapy. So the problem is that not every participant responds to aphasia therapy, and we know very little about what adjudicates between a good and a poor responder. So that's something that we're hoping to tackle in C-STAR. So if you look here, this is a figure from one of our earliest papers. This is from the Journal of Neuroscience in 2010. This is just a result of a aphasia treatment study, showing on the y-axis change scores in people's ability to name objects, so anomia. And what you see there is that everybody there on the left, so let's say 1 through 10, 11, 12, those folks do not respond to therapy at all. Actually, it may be a little bit worse. But if you go through 20, let's say through 26, those people show a very good response. So what we're trying to get at here is how can we upfront know who's a good or a poor responder? And I'll talk in a little bit about why we think that is very important. For me, working on aphasia recovery, one of the papers that I've always gone back to is this by my mentor, Audrey Holland, and colleagues. It was in the appeared in the Journal of Speech and Hearing Research back in 1989. I'm just going to read what they wrote here because I think it still very much applies. They said that there have been few prospective studies on the language recovery that occurs naturally during hospitalization for stroke and immediately thereafter. During this period, patients and their families are often perplexed about what happened, how their lives will be affected, what they can do, and what to expect. This immediate post-stroke period is also the time when patients may be medically unstable and difficult, if not impossible or inappropriate to test, when professionals working in the acute care hospitals are asked to evaluate patients, make prognostic judgments about recovery, and to begin to plan subsequent treatment. things haven't changed that much. We don't know so much about if somebody had a stroke yesterday, they have aphasia to a certain degree, where are they going to be a week from now, a month from now, a year from now, etc. So, like I said, very little is known about the factors related to recovery from aphasia. The general premise is that recovery of any kind of impairment following brain damage is that parts of the brain that remain functionally viable must adapt to both internal and external factors to drive recovery, or brain plasticity. The overall goal of C-STAR is to understand better the relationship between aphasia recovery and biographical, neurolinguistic, and neurophysiological factors. We want to inform both patients and their families, as well as clinicians working in the field. Now, you can imagine whenever you go to the doctor or you're admitted to the hospital, you want to know certainly what is the diagnosis. But also importantly, you want to know what is my prognosis. Where am I going to be at a given point in time? There's obvious reasons why we want to know that. We also want to inform clinicians because we think that perhaps not everybody is a good responder to impairment-based therapy for aphasia. What we would like to then suggest is that if you have very poor potential for improving your language, perhaps what you should be working on are compensatory strategies, counseling, how to live successfully with aphasia. And this is not a clear dichotomy. We're not saying that people should get either or, but perhaps the emphasis should be greater on impairment-based therapy in those who have a lot of potential for recovery, but for those who have less potential, maybe we should be working more with those people on compensation. So there are 4 aims in C-STAR, and each one of those aims is directly related to a given project. So there are 4 main projects that are tackled in the grant. The first one is to test whether contemporary models of speech processing can be used to predict response to aphasia treatment in chronic stroke patients. The second one is to examine the effects of transcranial direct current stimulation, tDCS. So remember that word, I'll come back to it quite a bit when I describe the second project, in relation to selective serotonin reuptake inhibitors, or SSRIs, aphasia treatment outcome in acute and subacute patients. So now we're talking about somebody who just had a stroke or is perhaps 2 or 3 weeks post-stroke. Aim 3 is to develop machine learning approaches to predict impairment and response to treatment in aphasic patients. And Aim 4 is to refine and extend current models of speech processing to better understand aphasic impairment and recovery. So now I'm just going to go very briefly through the 4 different projects. The first project is called Predicting Outcome of Language Rehabilitation, or POLAR. I am the project leader there. Dirk Den Ouden is my co-investigator, and the local project leader at MUSC is Leo Bonilla, who is one of my main collaborators. There are 3 specific aims. The first one is to identify biographical and cognitive linguistic factors that predict aphasia treatment outcome, and then to construct a predictive model of aphasia treatment success and make that model available online to clinicians. The second one is to test whether dual stream model, the one constructed by Hickok and Popple, improves outcome prediction beyond what can be inferred based on biographical and cognitive linguistic factors. And then test whether the dual stream model provides greater improvement in outcome prediction compared to the classical Wernicke-Lichtheim-Geschwind model. So that's the earliest model that we know of that describes how language is processed in the brain. The final one is to test whether measures of learning potential predict overall treatment outcome. 2 learning tasks will be administered at baseline. One will focus on language-related learning, whereas the other one will rely on language-independent processing. And what we want to know then is that can we predict how well somebody responds to language therapy based on their learning potential at baseline. A little bit here on the background of the project. The default model, the first model that we're tackling in Aim 1 is what we call here this default model. What we're simply trying to do there is to say, do factors such as age, sex, education, and also testing factors like level of impairment of different aspects of language, can we predict outcome accurately using that model? The alternative is that to add the dual stream model, simply looking at damage to specific regions of the brain, described there in the dual stream model at the bottom, does that statistically significantly improve our outcome? So does knowing something about where the brain damage is improve our ability to understand who is going to recover well and who is not. But as an alternative, we compare this to this old model, the Wernicke-Lichtman-Geschman model, to see whether there is a statistically significant difference between those two models. The plan here is to enroll 120 people with aphasia in aphasia therapy. In addition, we will test 30 more folks who have aphasia but who are not going to go through treatment. That's mainly for projects 3 and 4. We will collect and measure a large number of predictors, behavioral data, biographical data, neuroimaging data. Data collection is accomplished by 4 speech-language pathologists. We also have 2 postdoctoral fellows. This happens all at the University of South Carolina and MUSC in Charleston. There are 2 kinds of treatment foci. One is phonologically focused treatment, which will be administered for 3 weeks, 5 times a week, and then semantically focused treatment for 3 weeks, 5 times a week. And we randomize who gets semantic treatment first and who gets phonological treatment first. So this is a crossover design. The outcome factors are naming ability, or anomia, and discourse ability. Can we improve people's ability just to simply produce speech or discourse? What we're hoping to get at the end of these 5 years is a comprehensive model to guide aphasia treatment. So understand, perhaps there are some people that do better with semantically rather than phonologically focused treatment, or also possibly, can we adjudicate between people that overall show a good response as opposed to people that do not respond at all? The second project is stimulation of language in subacute stroke. Quite clever name there, short is SLISSE. The project leader there is R.G. Hillis. The first aim is to test the hypothesis that anodal tDCS, so now we're talking about low electrical current brain stimulation, plus a structural language therapy is associated with greater gains in accuracy in naming pictures, so targeting anomia, compared to sham tDCS. So that's the placebo condition, plus the same structured language therapy. So we're just looking at, can we improve language therapy outcome by adding tDCS in acute and subacute patients? 2 is a little bit more complex. It's to test the hypothesis that subacute anodal tDCS plus language therapy delivered by a computer in over 10 sessions is associated with greater gains in accuracy in naming pictures, so recovery from anomia in people with aphasia who have been taking selective serotonin reuptake inhibitors, or SSRIs, continuously since stroke onset, compared with the identical individuals and intervention individuals who have not been taking SSRIs. Without going into any details, and I know that RG, when she gives her talk, she's going to get into this in a little bit more detail. But selective serotonin reuptake inhibitors are thought to extend the optimal window of early brain reorganization following brain damage. And then finally, what RG wants to do is to test the hypothesis that anodal tDCS over non-lesioned areas of the brain modifies dynamic functional connectivity measured with resting state functional MRI, specifically between targeted brain regions and other nodes of the language network relative to the placebo condition. This is just a little bit of the background here. RG certainly has collected quite a bit of pilot data for the grant. This is from our previous studies, Baker et al. And Fridriksson et al., both published in the journal Stroke, simply showing that if you add tDCS in chronic patients to language therapy, the outcome, at least in smaller samples, the first study was 10 participants, the second one was 8, there seemed to be an improvement in overall ability to name and also in the reaction time of a name. What we're trying to do is to show whether this can work also in acute patients. This was all done in folks with chronic aphasia before. So the plan here is to conduct a randomized double-blind sham-controlled trial. We're going to enroll 40 acute subacute patients that will be randomized to 2 conditions. The first one is anodal tDCS plus computer-delivered speech and language treatment, or SALT. And we will compare that to the placebo condition. So half of those participants will be randomized to the placebo, where they will get sham tDCS, so it's not real brain stimulation, and they will also get the behavioral treatment. The anodal tDCS for sham will be applied for 3 weeks, 5 sessions per week, with a total of 15 stimulation sessions total, as in the ongoing CADE study. The CADE study is a clinical trial that we're conducting at USC and MUSC. We're up to 67 participants now. The goal is to get to 74 participants by the end of March. So we're close to finishing that study. Ultimately, what we want to do is to compare data from Project 2 to our clinical trial that will be finished shortly here in chronic folks. The outcome factor here is naming ability, so recovery from anomia, and also discourse ability. The product here is to provide pilot data to conduct a phase 3 trial. So if we get a positive result with these 40 participants, what we will then try to do is to support a phase 3 trial. Project 3, multi-modality neuroimaging to predict recovery from aphasia, is led by Chris Rorden. There are 4 specific aims. The first one is to develop multivariate methods to disentangle damage associated with specific behavioral impairments. So if you're dealing with aphasia, what you will notice is that it's not a single impairment. It's a multitude of impairments. There are things like phonology, grammatical processing, semantic, lexical retrieval, that all might be affected to different degrees. What we want to try to do, what Chris wants to do, is to try to adjudicate between the damage that is leading to the different kinds of behavioral impairments. This is very difficult to do because the different types of impairments correlate certainly within individual patients. The second aim is to develop novel statistical methods like machine learning algorithms to predict aphasia treatment outcome. The goal here is to collect data and look at, when we know what the impairment is in the acute phase, plus what the neuroimaging data are telling us, can we tell somebody where they're going to be at a given point in time post that assessment? This is the ultimate thing that I think most patients want to know, in addition to the diagnosis, is what's going to happen to me. Can I perhaps go back to work? Can I go home instead of going to a nursing home? So what is the future? So we want to give them some, we want to predict or help them understand what the future holds. The third one is, actually I got that wrong. That's the third one. So the second one is to predicting aphasia treatment outcome. So it's a little bit different. The third one was the one that I just talked about. So just like in polar, the second aim is then going to focus on, can we predict aphasia treatment outcome with the behavioral and neuroimaging data. And I'll talk in a little bit about how this is different from polar or the first project. The final aim, I'm not going to spend much time on it. It's very important to us that do neuroimaging in people with stroke. We're simply trying to look for new ways that we can better map the damage that we see using automated methods instead of having somebody hand draw lesions on the brain, which is the way that we quantify both the location and the extent of damage. So a little bit on the background here, what we want to know is whether we can use images like this. So knowing where the lesion location is, what you see here in the left hemisphere of this person, this dark shadow right there is the brain damage that occurred. You also see this in the rendered brain right here, so like a 3D look of the brain. We want to know whether the extent of this damage tells us something about recovery, both spontaneous recovery and how well somebody responds to treatment. In addition to that, we're going to use many different MRI modalities. For example, ETI. I know this looks like multicolored spaghetti, but what this is the white matter tracks showing by different colors indicating different directions of those white matter tracks. And if I were able to rotate this image, what you would see is that there's a big hole here in this brain where all the tracks have been removed. We want to understand, can we improve our prediction of outcome beyond simply looking at the lesion by looking at the remaining white matter tracks and how much of those are intact following stroke. We will also look at brain cerebral blood flow. That's what that is showing us. And then just looking at links between different areas of the brain. That may look a little bit abstract, but this is just a diagram on the left hemisphere on the left and right hemisphere on the right showing the links that are associated with different kinds of impairment in people with aphasia. So the plan here is to rely on data from polar and SLISSE. We will develop tools using existing data. We have a lot of data that we have accumulated through the years, through my research, through R.G. Hillis's research. We will refine tools in Project 3 as new data become available. And the product here is an algorithm to predict acute recovery and treatment-assisted recovery from aphasia. Unlike polar, Project 3 is agnostic to specific hypotheses. and relies on big data to aid prediction. So it's not specific hypotheses that are being tested, but rather what we want to see is if we put all of these data together, can we come up with a model that tells us something about long-term outcome. Finally, for Project 4, explaining aphasic impairment in the context of contemporary models of speech and language processing. This is led by Greg Hickok, who is at the University of California, Irvine. He has specific aims. Aim 1 is to map the neural organization of computational subcomponents of naming using both voxel-wise lesion symptom mapping and voxel-wise lesion perimeter mapping. In this aim, we will investigate the neural organization of the subcomponents of naming in stroke. The second one is to improve and extend contemporary models of naming and the neurobiological mappings. While the contemporary models are successful in accounting for the range of naming response types that have been fed into the models, aphasic projection deficits are rather more complex than standard operational definitions admit. For this reason, and in keeping with the overall goal of C-STAR, to bring together theory and practice, Aim 2 seeks to push the computational models more towards clinical reality. So not simply trying to understand what the impairment is, but also how does that relate to individual patients, for example, and what those patients are experiencing. The third aim is to assess the utility of model during diagnostics. We'll use computational modeling to predict aphasia treatment outcome. So this very much relates to the first project, Polar. The possibility we will assess in this aim is whether we can do better at classifying patients pre-treatment by measuring their functional deficit directly using functional computational models rather than relying on lesion-based proxy, as is the case with Polar. Because ultimately, if you're working in a rehab center, a lot of times you have no access to this neuroimaging data. What we want to be able to provide, if the neuroimaging data is helpful, great. Perhaps we can then find ways down the road make those data available to clinicians. But I think that a much more practical solution would be to provide models that don't rely on so much on the neuroimaging, but rather would rely on the behavioral testing and the biographical factors, and perhaps in this case, in Aim 3, to come up with a computational model, and then the clinician knows, here are the things that I need to test, I plug those things into the model, and he can tell me something about what is the rehab potential. Okay? So a little bit about Project 4. This is something that I swiped from a paper by Brent Walker and Greg Hickok. Came out this year. This is the SNP model, initially developed by Gary Dell and later refined by others like Myrna Schwartz at Moss Rehab. It's a model of naming, simply. And it has those semantic and phonological weights. What Grant and Greg have done is that they've added this extra component that you see there at the bottom, the auditory motor weight. And what they've come up with is what they call the semantic lexical auditory motor model architecture, or the SLAM model for short. And what they have shown is that they can model naming errors more accurately than the previous SNP model. Especially in folks who have conduction aphasia. So those folks make a lot of phonological errors as opposed to perhaps semantic errors. So the plan for Project 4 here is to rely on data from Project 1, so Polar. While data are accruing, rely on existing data from various labs to further develop the SLAM model. We're already doing this. And the product here is a neurocomputational model that predicts naming errors in aphasia as well as aphasia treatment outcome. We also have 3 cores, so we have these 4 main projects, and then we have what we call the service cores. The first one is the management core. That's led by myself. This very much relies on the Medical University of South Carolina for data management. The person who is in charge of data management for the whole project is Jordan Elm. She is a professor at MUSC. And she is one of the people included what is called the Data Coordination Unit. The Data Coordination Unit coordinates clinical trials across the country. It's the primary site for StrokeNet. StrokeNet is the NIDCD's stroke clinical trial network. And so what DCU does is that it coordinates data collection across multiple clinical trials. So what they're hoping to do there is to make those clinical trials certainly more successful by having centralized repository of data. What that does for us is very crucial in that the clinicians that work with us, the speech language pathologists, they do their behavioral testing and then they simply go online and they fill in their data into a website. And what that website then does is that it's encrypted, but it allows them to, it sort of leads them through the data. And it doesn't allow people to continue until all those data have been put in, or at least some of those data have been put in. Once that is done, all those data get then saved to a centralized database. That enables us to do this across different sites. So now we only have 2 sites. But you can imagine that for a clinical trial with, let's say, 100 different sites, this kind of a mechanism is crucial. What I really like about it is that they think about data management issues that we certainly never came up with. So it's very helpful. The clinical core is led by R.J. Hillis at Johns Hopkins. She oversees all aspects of clinical management across different centers. So how does that work, given that we have USC and MUSC also involved in data collection? We have weekly meetings, Friday mornings early, and what we do is that we simply meet using GoToMeeting and talk about the issues that have come up, talk about solutions, what's coming up, which essentially is like meeting in person. So that enables RJ to really be on top of the clinical activities of CSTAR. We found this works really well. The final and remaining core is the neuroimaging core. The leader there is Chris Rorden, and his co-investigator is Ludwig Desai, also here at USC. What Chris and Ludwig do is that they oversee all aspects of MRI data collection, processing, and storage. So we essentially have a centralized brain trust, if you will, which is Chris Rorden and others, that helps us set up all the neuroimaging across the different centers. So at Johns Hopkins, MUSC, and here, and then helps with setting up pre-processing pipelines, storing those data, and ultimately helping us get the data into analyzable format. So if you're not familiar with some of the latest developments of the software Chris Rorden has been coming up, I would point you to his website. His latest software, NIDCD, I think is absolutely revolutionary in analyzing patient data. We are still developing it, or Chris is, and Grigori and others. But I think that this is going to bring amazing benefits to CSTAR. So I want to talk a little bit about the research team. We have a lot of people. What you see there at the top are the folks at the University of South Carolina, the Medical University of South Carolina, UC Irvine, and finally Johns Hopkins University. The nice thing about a project like this is that you have multiple people that can provide input when different problems come up. So essentially what you have is a little bit of a brain trust working on similar projects with a similar theme. Obviously the projects have to be different. It can't all be the same project. But there is a centralized theme in what we're trying to accomplish. We're really trying to improve aphasia treatment outcome, and we're trying to improve our prediction of that outcome. I think that working with people like Greg Hickok, Chris Rorden, R.J. Hillis, and everybody else included there makes it so that what we are going to accomplish is not just complete those goals that we have, but also publish multiple other studies, because the amount of data that have been generated and are going to be generated in CSTAR is quite substantial. So this allows us to do a lot of very interesting stuff. I want to finally say that our CSTAR website should be coming up pretty soon. We've been working on it for a little bit here. If you want to find out more information about us, you can go to that website. You can see the URL right there. One of the things that is missing there at the top that we would like to add is a section for people with aphasia. Because ultimately what we want to do is to provide information about what we want to do to the community of folks with aphasia. Because if you think there's a chasm between research and clinical practice, you can only imagine that the divide between basic research or even clinically applied research and the actual survivors of stroke and people with aphasia is enormous. So what we want to do is that whenever we publish a new paper, we want to have the first author of that paper write up in layman's terms, what does this mean for somebody with aphasia? Because we want to communicate not only our findings to the scientific community and the clinical community to try to improve the actual treatment, but also so that participants with aphasia can advocate for themselves, for example. So that's going to be a big part of this. I don't really know how long I went, but I'm going to stop at that, and if you have any questions, I'd be happy to take those. Thanks. Interesting to see how that works. Okay. Should be. Yep. From.
Just if you check your audio, we should be able to use it from your audio on your
The problem is that the mouse has disappeared. There we go. I'm going to have to switch this. Let's see. The mouse only wants to work on the screen. This is definitely a problem.
Yeah, there you
go. All right.
You go to your own audio and click on the speaker.
Okay. Here. Okay. All right. This? Yep, you can see it. Okay. All right. Anybody have any questions? John?
Good idea, but you name like 2 or 3 of interventions.
Yeah.
I presume there's hundreds of different interventions.
Yeah.
So are the ones you choose ones in particular that you think are more general, or
yes.
When you do these 2 and do all this massive stuff, I was not going to predict how Joe Blow's intervention number 7.
That's an excellent question. So we did exactly what you suggested. We are picking very generalized interventions, and we're dividing them up into either phonologically based or semantically based. And what we've done is that we've taken 3 approaches from the field, if you will, 3 that are semantically based and 3 that are phonologically based, and we have sort of grouped those together to make up the 2 phases. What we're trying to say is not that we're proving that a given therapy works, but rather that if you give somebody like a hodgepodge of approaches that focus on semantic processing, we can compare that if we focus on something else that we know is very much impaired in aphasia, which is phonological processing. There are different ways that we could have done this. But the question is, should you pick one specific treatment and only focus on that, because you're then putting a lot of eggs in one basket? That's a great question.
Just along that same line, are the links between the real specific to some idiosyncratic, idiopathic etiology, or are they more general, like one brain area will affect several things? How does that?
Yes. So yes and no. So I would say that what we're trying to do is that we can measure the initial impairment. We can try to adjudicate between the different types of impairment that that person has simply based on the brain damage itself. What we're hoping to do then with the other MRI modalities to give us some extra information about, what is the residual brain doing, if you will, looking at things like functional connectivity, which we are already showing with a couple of the people working in our lab, Bree Stark and Grigory Yorganov, that you can predict errors, for example, in aphasia much better by looking at the residual connectivity rather than the actual damage. Stacy?
Along those same lines, it appears that interventions are not standardized, but more general to the impairment. Is there any standardization as far as dosage besides 3 times a week? Is there a time or repetition or intensity dosage?
So there is no clinically standardized dosage. And what we did was that we simply looked at what is the standard dosage in the United States? What is the average dosage that you get? Because there are so many different ways that you can slice this. We could have gone for a dosage study, but then like half of the other stuff we would have had to cancel. So I could probably write a new P50 grant simply focused on something like dosage. So I thought that just to play it safe, what we looked at was if you have a stroke in the United States, you have aphasia, how much therapy are you likely to get? And we went with that.
And that's for the therapist that will be delivering the treatment.
Yes. Yes. We actually started data collection already, and it's going really well. We have speech pathologists on site that are already implementing that type of thing. It looks very promising. All right, any other questions? All right. I don't see them. Can you read them?
I don't see any chats, but if someone has
The question was, what is that big face?
I think if people have questions, we would be hearing them right now. So.
Yep. Okay. All right. I guess we're good then. All right. Thank you, guys. I appreciate it. And just again, we are planning to put this lecture and every C-STAR lecture subsequently on our website. So I would say watch that space. Thanks.